Hello and welcome back to another Dr-Tutor video. Today we're going to be discussing the A-level topic of cellular control in biology. This video is based on the OCR specification but has been adapted slightly so that it is a bit more suitable to other specifications as well. First we have to discuss mutations, and we have two key types of mutation: point mutations and insertion/deletion mutations, or indel mutations. Point mutations are where a base pair may be replaced and substituted, and indel mutations can cause a frame shift as one or more nucleotides is essentially inserted or deleted. In terms of point mutations, we have three further types. Silent mutations are where, despite a mutation being caused, there's no change to the primary structure of the protein, and thus there's no significant effect of this mutation. This is possible because genetic code is degenerate, and as a result, multiple triplets will code for the same amino acid. This is key in playing a role in decreasing the effect of mutations, as they don't always thus lead to a change in the sequence of amino acids in a protein, and thus they don't necessarily affect the primary structure of the protein and thus the function. If there is a change in the sequence that leads to an altered amino acid sequence in the protein, this is a missense mutation. Such a change may cause alterations to the secondary and tertiary structures and cause the protein to be ineffective or stop it from carrying out its function. An example of the result of missense mutations for example, could be sickle cell anemia. Finally, we have nonsense mutations. This is when a mutation alters the base sequence such that a stop codon or stop triplet is formed. This will result in a short protein that is unlikely to function. Moving on to insertion-deletion mutations. If a base pair is inserted into a gene or deleted, it will cause a frame shift, as DNA is non-overlapping. This results in all subsequent triplets being altered. This means the subsequent protein formed from this gene is highly likely to be severely disrupted, and the primary, secondary, and tertiary protein structure will thus be severely altered. Such a protein will most likely not function correctly, if at all. It is, however, important to note that sometimes a triplet may be added or deleted, and this results in the additional removal of an entire amino acid, and therefore not necessarily a frame shift. We can also have cases where there's a repeating triplet, and at meiosis the number of repeats continually increases. This is the cause of Huntington's disease, for example. You need to understand the concept of gene regulation. This is required as some genes will only be synthesized under specific conditions or environments, while others won't. DNA has introns and exons. Introns are non-coding regions of DNA, while exons are coding regions of DNA. It's important to understand that we also have an operon, which is a group of genes which function as a transcription unit. This will make more sense shortly. We have a few examples of gene regulation in real life. First is the lac operon. The lac operon is found in bacteria, which would normally metabolize glucose, but if glucose is absent, they still have to respire, and so they can use lactose. Thus, in the absence of glucose and the presence of lactose, two enzymes are synthesized: lactose permease, which enables lactose to get into bacterial cells, and B-galactosidase, which hydrolyzes lactose to produce glucose and galactose. The lac operon consists of a length of DNA with an operator region, LAC-O, next to two genes that are LAC-Z and LAC-Y. These two enzymes code for the two enzymes I've already mentioned. Next to the operator region is the promoter region. This is the site which RNA polymerase must bind to begin transcription. Further upstream of the promoter region is the regulator gene. This codes for a repressor protein for the operator region. This means that if the repressor protein is produced, it will bind to LAC-O and prevent the transcription of LAC-Z and LAC-Y genes. This means that lactose cannot be metabolized as the genes are essentially switched off. This is all facilitated as the repressor protein has a structure that is complementary to the operator region. In the presence of lactose, when all glucose has been used, the lactose will bind to the repressor protein molecules, which causes a conformational change in the shape of those molecules and makes it no longer complementary to the operator region, so it can no longer bind to the region. This means that RNA polymerase can bind to the promoter region and transcribe the genes and thus synthesize the enzymes that are required to hydrolyze lactose and thus provide glucose for respiration. Crucially, however, the lac operon is only found in prokaryotes. In eukaryotes, we have a different system. Now, I'm not going to go through eukaryotic gene control in too much detail, as the amount you need to know on this varies based on your specification. However, the fundamentals are that the basis of specialization is that not all genes are being expressed in all cells. They all share expression of common housekeeping genes, however. Gene expression is controlled by transcription factors, which are short non-coding RNA that combine with a specific site on a length of DNA and inhibit or activate transcription of the gene or genes. A transcription factor will bind to its specific promoter gene. After binding, they either aid or inhibit RNA polymerase binding. They're essential for the regulation of gene expression and regulating the cell cycle. An example would be tumor suppressor genes and proto-oncogenes, which help regulate cell division, and mutations could lead to cancer. When it comes to gene regulation, we have post-transcriptional and post-translational gene regulation. In post-transcriptional gene regulation, all DNA, intron or exon, is transcribed, and the product is primary mRNA. This undergoes a form of editing to remove the introns, or the lengths of mRNA, which correspond to the introns on the DNA. The mRNA exons, post-processing, are then joined together. This process will typically use the enzyme endonuclease. In post-translational gene regulation, activation of proteins is primarily through phosphorylation. So what typically happens is a signaling molecule, such as protein, binds to a receptor on the plasma cell membrane, which is associated with what's called a G protein. This binding activates the G protein, which in turn activates adenyl cyclase. Adenyl cyclase catalyzes the formation of many molecules of CAMP from ATP. The CAMP, which is cyclic AMP, activates pKa, which is protein kinase A. This then catalyzes the phosphorylation of proteins which utilize ATP through hydrolysis of ATP, and the phosphorylation then activates many enzymes in the cytoplasm. These could be those that convert glycogen to glucose, for example, etc. Protein kinase A then can phosphorylate another protein, and so on, and this may enter the nucleus and act as a transcription factor. Moving on, we need to understand genetic control over body plan development. We have homeotic genes, which are involved in controlling the anatomical development or morphogenesis of an organism, so all structures develop correctly. These often contain homeobox sequences or homeobox genes, which are sequences involved in regulating the patterns of anatomical development in fungi, animals, and plants. The length of this chain, including only the introns, is 180 DNA base pairs, which thus codes for a 60 amino acid sequence that is known as a homeodomain sequence. This sequence can fold into a shape and bind to DNA to regulate the transcription of adjacent genes and thus transcriptional factors. The shape they form is called the HTH, as it contains 2 alpha helixes and a turn in between. Homeobox genes are very similar and highly conserved. Molecular evidence shows that homeobox genes were found millions of years ago and shows that they arose in early ancestors and have been conserved since then. The sequence is similar in all animals studied to date. A quote-unquote subset of these is Hox genes, which are found only in animals. Hox genes control which body parts grow where. In mutated Hox genes, there can be growth abnormalities. Hox genes can also be involved in stimulating mitosis or apoptosis. Apoptosis is programmed cell death, and the process is seen in the diagram. Essentially, enzymes break down the cytoskeleton of the cell, and the cytoplasm becomes dense with tightly packed organelles. The cell surface membrane changes, and small protrusions emerge, which we call blebs. Chromatin then condenses, and the nuclear envelope breaks and forms DNA fragments. Then the cell breaks into fragments that are ingested by phagocytic cells, so debris can't damage any other cells. The process of mitosis we should have already learnt in previous topics, and if you're unsure on it, you can check my notes on dr-tutor.co.uk. Now, this is controlled by numerous cell signals which may be released due to internal stimuli such as stress, as well as signaling molecules such as cytokines, and certain hormones and growth factors like nitric oxide, etc. Nitric oxide acts to make the membrane more permeable to H+ ions, which makes the proton gradient essentially lessen. Equally, proteins may bind to apoptosis inhibitor proteins, allowing apoptosis to occur. Apoptosis is key in the development of limbs and enabling digits to separate from each other. It's also important in removing ineffective or harmful lymphocytes during the immune system's development. However, it's important to note that the rate of cell death should be equal to the rate of cells produced, as if it's more there will be cell loss and degeneration, and if it's less there will be potential tumours. On average, cells will undergo mitosis about 50 times before dying, and this is what's known as the Hayflick constant. Now that brings us to the end of today's video on cellular control. I have covered the main points in this topic, however based on your specification there will be certain points which haven't been covered in as much detail, and some specific points that may not have been covered at all. If you want one-on-one help in improving your grade or covering specific points based on your specification, you can book tuition with me using my website Dr-Tutor.co.uk, and we'll also go over exam technique and past paper questions and make sure you know the best way to study for yourself. If you're interested, leave a comment or head to my website to book a free consultation meeting. If you're applying for medicine, I also offer interview preparation and UCAT preparation sessions, as well as personal statement reviews. I hope you found today's video useful, and if you did, make sure to leave a like and subscribe for more weekly videos. Otherwise, thank you for listening and see you next week.